We have studied different types of muscles. We have studied skeletal muscles, the general characteristics of skeletal muscles, smooth muscles, and choric muscles. And now we are going to study in detail the structure of skeletal muscles. And later we are going to study the mechanism of muscle contraction also. Before that, we are going to study what is the detailed structure of skeletal muscle. Starting right from the beginning, this is a muscle. This is a muscle. And this muscle actually is made up of bundles of muscle fibers. And actually a muscle is protected over by an external connective layer, and that connective membrane or connective layer is called epimysium. And epimysium covers to the structure of muscles externally. And epimysium, within epimysium there are several kind of bundles of the muscle present. And these are said to be the bundles. These are bundles that are present in muscle. And to each this bundle we have taken out, and this is extended structure of that structure. This is called fascicle. To all these structures we say these are called fascicles. So a muscle is made up of many fascicles which are externally protected over by epimysium. And if we study the structure of fascicle, fascicle itself is externally protected, externally covered over by a connective layer, and that layer is called perimysium. To the membrane of a fascicle, the layer of a fascicle we say is called perimysium. And what actually fascicle contains? See, within fascicle there are many fibers present, and these are called muscle fibers. So a fascicle contains many muscle fibers, and to one muscle fiber we have extended out from fascicle. This is called one muscle fiber. And as we say, a muscle fiber is present. There are many muscle fibers that are present in fascicle, and there are many fascicles that are present in muscles. And this way, muscle is externally protected by epimysium, and within muscle there are many fascicles, and we have taken one fascicle out, and the fascicle is covered over externally by perimysium. And within one fascicle there are many muscle fibers present, and this is a one muscle fiber that we have taken out, and this one muscle fiber is also protected, is also covered over externally by a membrane that's called endomysium. And this is how is an arrangement of epimysium, perimysium, and endomysium. Endomysium covers over externally to actual structure of muscle fiber. And what actually muscle fiber is? What is present in muscle fiber? What are the functions and other characteristics of muscle fibers? We have elongated, we have elaborated the structure of muscle fiber a bit broader that you can understand it very easily. So this is a one muscle fiber. Muscle fiber is externally protected, externally covered over by a layer called endomysium. And inside endomysium there is a membrane. Muscle fiber actually is in a cellular structure which actually works as a functional unit of muscle. Muscle fiber can be considered as cells of muscles. When we consider it as in a cellular structure, it is externally protected by endomysium, and under endomysium there is a membrane present that is called sarcolemma. This actually is in a cytoplasm that contains, this is actually a membrane that encloses the whole cytoplasm, sarcoplasm of a muscle fiber. So that layer, this layer is called sarcolemma, and it actually is a layer which would definitely receive the impulses also, and that will later conduct the impulse through transverse and T-tubules throughout a whole structure of muscle fiber, and that would later help sarcomeres to contract and relax muscle. So this is called sarcolemma, and within this is a whole structure of cytoplasm, and to the cytoplasm of muscle fiber we consider it's called sarcoplasm. And the sarcoplasm, which is rich in a specific kind of a endoplasmic reticulum called sarcoplasmic reticulum, that releases calcium in order to ensure the contractions of muscles. So this is called sarcoplasm within one muscle fiber. See, these are structures present. This is a structure, this is another structure, and to this is structure. These structures are called myofibrils. We say these are said to be, these are myofibrils. And each myofibril is basically made up of specific units of the filaments, and to these units of filaments we say they are called sarcomeres. These are called sarcomeres. And sarcomeres combine to make a myofibrils. And when myofibrils combine, they make one muscle fiber. And when many muscle fibers combine, they make fascicle. And when many fascicles combine, they make the actual structure of muscle. And if we detail what actually sarcomere is, and how actually it works, and what are different kinds of filaments present in it, we are going to draw a detailed diagram of a sarcoma so that we can study the different kinds of filaments, thin and thick filaments that are present in this basic structural unit called sarcomere. And before starting sarcomeres, I must explain something, another important thing. This sarcolemma that's present externally at the skull side, cell membrane of a muscle fiber, that can form a tubules-like structures inside. That can form a transverse tubules-like structures like this. These are called transverse tubules-like structures called T-tubules. And these T-tubules can penetrate to a muscle fiber deep throughout so that the impulse message can travel to all the kinds of myofibrils, and via myofibrils the message can transfer to sarcomeres, and sarcomeres can have calcium, and thus they can slide over each other and contraction starts. So in order to conduct the message throughout a muscle fiber, sarcolemma invaginates inside. It forms the tubules-like structures that are called transverse tubules. These are called transverse tubules or T-tubules. That actually are invaginations of sarcolemma within muscle fiber, and that will help conduct impulse. So we are going to study the structure of sarcoma in detail, and if we draw the structure of sarcoma, so what actually sarcoma is, and what filament sarcoma is made up of. So each sarcoma has an outer boundaries, outer linings, outer ridges, and outer sides. And to these sides we say this is said to be a Z line. To this outer boundary of a sarcoma, we consider this is what? This is called Z line. And these Z lines actually limit, they determine the boundaries and sides of each sarcomere. And within these Z lines actually is the structure of sarcomere, which is made up of different kinds of filaments. See, within Z lines, a sarcomere has different regions. Say these are filaments. And to these filaments we say these are thin filaments, and these are actin filaments. And actin filaments are extending from Z lines, and they extend towards the center of sarcomere. And these actin filaments, they make a region of a sarcoma where only thin filaments are present, and it becomes a region of a sarcoma. To that we say it is called I-band. The region of sarcoma where only actin filaments are present, this is said to be a region of a sarcoma. It's called I-band. And from I-bands inside is a region of a sarcoma. That region is called A-band. This extends from I-band from this side and I-band from this side. This whole inner middle region of a sarcoma is regarded as an A-band, which actually is a region where filaments would slide over each other. And this actually is a site, this actually is a place which never increases or decreases, but this is a place where actins and myosins will slide over each other. And in an A-band, there is a specific region, the central region, where only a thick filament is present. This thick filament is present. And to that region, which is actually a region of A-band, this is called H-zone. Basically, there are two important filaments that we are discussing. Thin filaments, these are called thin filaments, and these filaments are called thick filaments. Thin filaments are actin filaments, and thick filaments are called myosin filaments. And the region of sarcomere where only myosin are present is regarded as an H zone. Whereas A band is actually a region where H zone is at center and it is present on both sides, where filaments such as actin and myosins both are present, or they would overlap in this region that is actually called A band. And at the center of the myosin filament or H zone, we have a line that's called M line. This actually is a structure of a sarcoma. Now you must be understanding these are called Z lines, and from Z lines there are I bands, and I bands are bands where only actin filaments are present, and then there is a region called A band, and A band extends from the I band from this side to the I band from this side, and this whole region is called A band, and within A band there is a region where only myosin filament is present, and to this structure we say, to this zone we say it's called H zone, and center of it is regarded as an M line. This is a structure of sarcoma. Sarcomere is actually made up of different kinds of filaments. By words we are saying these are called actins, and we are saying they are tropomyosins, and they are troponins, and they are called myosins and etc. What actually these filaments are, actually these are proteinous filaments, and how actually they are composed, what are their units, and how they are formed. We are going to make a little detail of these filaments, and so that we must be knowing everything about sarcomeres and their filaments, and later we can easily study how muscle contracts and what actually is the mechanism of muscle contraction. So actin, we say that sarcomere is made up of actin and myosins. This is a actin. And actin is made up of small subunits, these are called G-actins. And when G-actins combine, they make a fibrous actin called F-actin, and two chains of F-actin. These are two chains of F-actin. When combined, they arrange in a helical fashion, and this helix-like arrangement of two chains of F-actin make actually a filament. To this filament we consider it's called actin filament. So this is one important thin filament that is present in sarcoma. To this thin filament we say it is actually made up of this is called actin filament. Or similarly, there is another filament that is again a helix-like structure. This is true helix-like structure. This is regarded as a tropomyosin. And tropomyosin actually is an actin myosin inhibitor, because it can inhibit the contraction of muscle. When it hides the sites of actin, cross bridges can't get attached, and muscle's contraction is inhibited and muscle is relaxed. Actually, this filament is called tropomyosin. And on this tropomyosin, at different interval, there is another small protein, small filament present, and to that filament we say it's actually called troponin. This is a structure of a troponin. This is the structure of a troponin. And actually troponin is a filament that is present on tropomyosin. It's present on tropomyosin. A troponin is basically made up of three units. It has TNL unit, which actually is an inhibitor site which makes troponin able to bind with actin. And it has an N, TNTN. And this TNT site or end of a troponin makes troponin able to bind with a tropomyosin, and it has one end that's called TNC, and where troponin can bind with a calcium. To get the kind of at the time of contraction of muscles, calcium is to bind to a troponin, and when calcium binds to a troponin, it actually twists to the structure of tropomyosin, and when tropomyosin twists, it actually exposes the sites of actin, and then only can cross bridges attach and muscle can contract. This is how the mechanism of muscle contraction would work. So before that, you must be understanding what is the structure of these filaments. So this is called tropomyosin, and on tropomyosin there is a filament present called troponin, and troponin has these three units or components. The last filament is a thick filament. This filament which is called thick filament. And this thick filament actually is made up of polypeptides, polypeptide subunits. 6 polypeptide subunits combine to make a basic molecule, and to that molecule we say it's called myosin molecule. And this myosin molecule is having long tail and 2 bulbous heads to cross bridges. This is head and this is called long tail. It is actually made up of 6 polypeptide chains. Such myosin molecules are to combine to make a thick structure of myosin. And around 300, these myosin molecules, combine to make a basic thick structure of a myosin filament, which actually appears like this. You see, this is said to be a myosin filament, and this myosin filament is actually made up of 300 myosin molecules, where they have a long tail and bulbous heads extending that would act as a cross bridges, and they would attach to the sites of actin, and they would actually help in a contraction of muscles. So this is a one unit. This is called myosin filament, and on the other there is another complex, which actually is a complex that is present around this myosin, which is exposing or hiding sites of actin, and thus either helping muscles contract or relax. Now see, these are two chains of actin that are in blue color, and these two helical chains of actin, on them have a structure of a helix-like arrangement, which is in a green marker. This green color shows another filament that is called tropomyosin, and on this tropomyosin there is another filament present that is called troponin, and this is how they all make another complex which is present around this structure of a myosin, which actually make a structure of sarcomere, where these structures of actin along with tropomyosins and troponins, and at the center of myosin they are making a structure that would actually slide over, and when these filaments will slide over, sarcomere will contract, and a sarcomere when will contract, it will contract the myofibers, and when myofibers contract, they will contract the muscle fiber, and muscle fiber contracts, that contracts the fascicles, and when fascicles contracts, the over structure of muscle contracts and it becomes short and thick. How it contracts is the mechanism that I am going to explain in a next video. Subscribe my channel, share it with your friends, and let's study together.